Welcome one and all. It is Wednesday, June 10th, 2020, and we are kicking off the very first of a series of IPM tick webinars offered through the Northeastern IPM Center. I am Jana Hexter and I will be hosting the webinars, and we have a wonderful lineup for you all throughout the summer. We're still putting some in place, but they will be going into the fall. And the first person that we have to introduce to you today is Dr. Kirby Stafford. And Kirby, if you would like to say hello, that would be great.
Good morning, everyone.
Good morning. Great. And we have, Kirby is actually moving the slides initially, so I will ask him to move to the next slide while we just set up some prep. All right, so there will be a recording of this webinar available within about a week. We do edit it, so it's going to be at this link. And I will also email everyone who is registered for this webinar, and you'll get a copy as soon as it's ready. I'll send you an email. So if you don't hear from us by the end of next week, just shoot me an email in case we have an incorrect address for you or something like that. And the next slide, please. Okay, so what we would like to do is to use the Q&A feature rather than the chat feature for asking questions. I know some webinars use the chat feature, and we like to use the Q&A because it helps us to be able to keep track of the questions. Sometimes people ask, the three people ask the same kind of question, and so we can see that. And then we can ask, we have some breaks scheduled. Kirby's going to be presenting some material, and then we are going to take some breaks and ask for some questions. And so it helps us to be able to keep track. You can also ask questions anonymously if you're more comfortable doing that. And so we won't be monitoring the chat feature, but we will be monitoring the Q&A feature. And the next slide, please. Oh, I seem to have, oops.
I did advance it.
Okay, all right. I seem to have lost that, so I'm just going to, for some reason, it is not on my screen anymore. Okay, it's gone back to the first one for me. So I think the next slide is me to introduce Kirby. I will do that. So Dr. Kirby Stafford works at the, is the chief scientist at the Connecticut Experiment Station. He joined in 1987, and his research is in the area of ecology, distribution, and control of the black-legged tick, Ixodes, scapularis, and more recently the Lone Star tick, with a major focus on natural, biological, and integrated tick control. And he's actually going to be doing two presentations for us. Today he's going to be talking about strategies and barriers to the prevention of tick-borne disease. And then at the end of the summer, in fall, I think in October, he's agreed to come back and talk to us about leaf litter control and snow removal techniques to help with ticks. So we're super excited that he's here and presenting for us. And in the next slide, we have some questions for you. We can bring up the poll, please, Kevin. That would be great. All right, great. So you'll see here that we have a few questions for you. This helps us to understand who's on the call, the level of skills and understanding on the call, and then also for us to be able to ask similar questions at the end to see what the impact may have been. So this is not class, this is not school. There are no right and wrong answers. It's just your best guesses. And we'll give you a couple of minutes for you to answer those questions, and then we'll share the results. All right. So it looks like most of the people on the call are somewhat or moderately knowledgeable, and we have one very knowledgeable person. That's wonderful. And the incidence of tick-borne, knowledge of incidence of tick-borne diseases is about the same. So we pretty much, if you look at these that most people would put themselves in somewhat knowledgeable or moderately knowledgeable. And so that helps us to know, or Kirby to know, how to gauge the conversation today. And thank you for doing that for us. So with that, I will turn it over to Kirby for his presentation.
Okay, thank you, Jenna. I'd like to start my talk with this quote from Dan Sonnenshine's Volume 2 textbook on biology of ticks, where he noted that few agricultural or health problems confronting human societies have proved as intractable as the control of ticks and the many diseases they transmit. And indeed, there are a lot of diseases. And today, to kind of address that question, we're going to quickly go have an overview of tick-borne pathogens, briefly highlight tick surveillance, get into more detail on tick integrated pest management and their various components. Host-targeted tick control is one of the major focuses of that. Kind of highlight some of the challenges that we face to effective public tick control and future webinars. I will be going through some of this relatively quickly, but bear in mind this is being recorded, so you would have the option of watching it again. And I will also refer to a few publications as I go through here that are open access, so you can easily follow up for more detail. So let's talk about the issue that we're dealing with in terms of the tick-borne pathogens. This is a recently published timeline where we look at from the first initial discovery of Rocky Mountain spotted fever in the late 1800s and the early 1900s. You can see a steady discovery of a variety of tick-borne pathogens that cause human disease, all the way up to Borrelia burgdorferi, the causal agent for Lyme disease in 1982. And since then, you'll note that the discovery of tick-borne pathogens has accelerated with a variety of agents found in amblyoma species ticks and Ixodes species ticks, as well as dermacenter species ticks. So we're seeing a rise in the total number of tick-borne diseases here in the United States, reported to the Centers for Disease Control and Prevention. And bear in mind, this probably only represents maybe 10% of the actual diagnosed cases. And so we're nearly 60,000 cases that were reported in 2017. And of course, the bulk of that is Lyme disease. And as you can see, from this 22-year expansion in the Lyme disease case distribution in the Northeast and upper Midwest from 1996, when we had a little over 16,000 reported cases, to 2018, where we were getting almost up to 34,000 reported cases. A lot of that has to do with the geographic spread of the black-legged tick, as putting more people at risk of exposure to the tick and getting Lyme disease. So Lyme disease does continue to be the major vector-borne disease in the United States and tick-borne disease. And we're talking, again, remember I said only about 10% of the diagnosed cases are actually end up probably being reported. So the true incidence is probably somewhere around 330 to 430,000 cases of Lyme disease every year, but which represented 68% of the total in 2017. But interestingly, you'll see that other tick-borne diseases is starting to comprise a bigger part of that pie, 27% of the tick-associated diseases that we have. So the major ticks here in the Northeast that we're dealing with are 3 species that commonly bite humans. Even though there's about 17 species of ticks in the Northeastern United States, most do not actually feed on humans. So the 3 common species, of course, is our black-legged tick, or as many know it, the deer tick, Ixodes scapularis, the American dog tick, Dermacenter variabilis, and increasingly the lone star tick, Amblyoma americanum. And I'll be talking briefly a little more about that one in a moment. We do get records of the woodchuck tick, Ixodes cookii, from humans, as well as occasionally another tick called Ixodes dentatus, which is kind of a rabbit tick, as well as occasional human cases of tick bite from Rhiphocephalus sanguineus, the brown dog tick. What is really of interest is the Asian longhorn tick, Haemaphysalis longicornis, and I'll talk briefly about this. This is a new exotic invasive tick that has come in here into the United States. Now, all of these ticks that I'm talking about today are three-host ticks, meaning that you have three active stages: larva, nymph, and adult, and each stage feeds on a different host animal. And what I've diagrammed here is the life cycle of Ixodes scapularis, the black-legged tick. So the larvae feed mainly on rodents and birds, although in this case they will feed on any animal at all, including us humans. Once they feed, they drop off and become nymphal ticks. The nymphal ticks have to host seek and find another host animal again. Again, mainly rodents and birds, but again, the nymphal ticks will feed on any animal, including humans. And then once they feed, they drop off and become adult ticks, male and female, which feed on medium to large animal hosts. They're not feeding on the mice or the birds, but primarily white-tailed deer. That's the principal host for the black-legged tick adults. And it's also the primary host for all stages of the Lone Star tick. So each female tick that engorges on a white-tailed deer will drop off and lay about 2,000 eggs, starting the life cycle over again. Now, those different stages are active at different parts of the year. And what I've done here is overlaid the seasonal activity of Ixodes scapularis in the Northeast, slightly different in the upper Midwest, with reported, confirmed, and probable cases of Lyme disease by week of disease onset. And if you'll notice here that the peak of Lyme cases occur in the summer months between May and August, particularly June and July. And that is a time when the nymphal stage of the black-legged tick is most active, so which is shown in the yellow curve that you see here. So if we run through the life cycle here, eggs are laid by female ticks primarily in the early spring, May. Larval ticks hatch from those in the mid-May, from middle July into August. So August is the peak month of larval black-legged tick activity. Once those ticks feed and drop off, they molt to nymphs, and they actually overwinter as nymphs and then show up the following year as nymphs. So the nymphs that you see there in the middle of the curve were the previous year's larval ticks. Once those nymphal ticks in the summer here now feed, they drop off, molt to adults that show up in the fall. They'll be active even in the winter months when it's warm. They do not hibernate, and you get a smaller peak of adult tick activity in the spring. But the main thing I want to emphasize here is the primary exposure. In most cases, Lyme disease occur in the summer months and are associated with the nymphal stage of the tick. How do we know when and where these ticks are active? Well, that's due to a variety of approaches on tick surveillance. There are two types. One is the passive and the other is active. Passive tick surveillance is when ticks are actually submitted to the Extension Service or to universities or other institutions that will accept and identify and sometimes test ticks for the public. Active tick surveillance means researchers are going out and actively sampling for the ticks, primarily by dragging for them. And you see illustrations here of researchers dragging for ticks. And this allows us to monitor changes in the distribution and abundance of ticks, seasonal activity, and the presence and prevalence of tick-borne pathogens in these ticks to provide more actionable evidence-based information, not only to clinicians and doctors, but you all as well, and that to give you better information on when and where the risk is. So now, as I talked about a little earlier, I was going to go into Lone Star tick a little bit more because this is an emerging issue here for the Northeast. The Lone Star tick is responsible for 90 to 95% of the tick bites in the southeastern United States, and it has been steadily moving north. And you can see the map showing the approximate distribution there on the far right. There's a different suite of disease pathogens that are associated with the Lone Star tick, a couple of viruses, the Bourbon virus, Heartland virus. The main pathogen associated with this tick is Ehrlichia chaffiensis and Ehrlichia ewingii, which causes human ehrlichiosis. But we also have a few other diseases as well, like tularemia and spotted fever group, rickettsia. But the one that tends to get a lot of attention is red meat allergy, which has been associated with the bite of this tick. This is an allergy to red meat, also called alpha-gal syndrome. It's a sugar, and that can range from mild gut upset to hives to full anaphylactic shock. So something that we're watching as well. So we've been actually monitoring this expansion of the Lone Star tick into the northeastern United States. It first moved into Long Island, New York in the early 1990s. And then we have found that the adults of this tick can survive in Connecticut and even up in coastal Maine. And so our current environmental and climate conditions, which are somewhat moderate maritime climates, favor the establishment and expansion of the Lone Star tick along the New England coast. So it's been now become established on the islands off the Cape, Cape Cod. We're increasingly finding more of these ticks here in southern Connecticut, even though the numbers are still relatively small, except for one island off coastal Connecticut where back in 2017, we discovered a well-established, very dense population of the tick. So it is expanding it. And we published actually a notice to the medical community called Bracing for the Worst, Range Expansion of the Lone Star tick in the northeastern United States, because physicians need to be aware of some of these other diseases that'll show up other than just Lyme disease and those associated with the black-legged tick. The Asian longhorn tick was discovered on a sheep at a farm in Hunterdon County, New Jersey, way back in 2017. This exotic East Asian tick is considered a serious pest to livestock, including cattle, horses, sheep, and goats. And it will attack pets, particularly dogs, wildlife, and only occasionally humans. The unique thing about this tick is it reproduces parthenogenetically. No male tick is required, and it can build up really high populations very quickly. It is known vector for a number of human and animal pathogens in its native range in parts of China and the Koreas and Japan. It became established back in the late 1800s and early 1900s in Australia and New Zealand, where it's mainly a livestock pest. So the story of this particular tick is still emerging. It's been found in 12 US states so far, as you can see here on the map, ranging from Tennessee up to Connecticut, as well as one detection way out in Arkansas. We expect this distribution to expand as our surveillance efforts increase. Recent lab studies have found that it is not a vector for Borrelia burgdorferi, the agent of Lyme disease, but in the lab it was found capable of transmitting the agent for Rocky Mountain spotted fever, Rickettsia rickettsii. At this point, it looks like it's mainly going to be a veterinary concern, and it doesn't seem to really like humans all that much. At least here in the United States. So this is an evolving story. So we'll see what happens as time progresses and the surveillance activities continue. So when we think of ticks as vectors, I mean, they're different than mosquitoes. So there's kind of two aspects to look at this. There's increasing evidence, as I quote from this paper, from detailed analysis that rapid changes in the incidence of tick-borne diseases are driven as much, if not more, by human behavior that determines the exposure to infected ticks than by tick population biology that determines the abundance of infected ticks. But conversely, habitat diversity, environmental factors influence survival and tick activity, and the geographic distribution of ticks impacts the risk of tick-borne disease. And it really is both of these elements that come in to defining what your risk of a tick-borne disease are. Because remember, ticks are found in wooded and successional habitats in relatively high numbers. And the degree with which they're infected and the tick-borne disease incidence is very endemic. It tends to be non-focal. I mean, the ticks are broadly distributed. They don't fly. So you actually have to enter or live in a tick habitat and become exposed. And increasingly, we've seen homes built into the forested areas, which is essentially tick and host habitat. So, you know, a lot of people ask how many are infected, and that's an important parameter, but infection prevalence is only somewhat predictive of your transmission risk. Actually, tick abundance and the number of tick bites you receive is more important of your chance of encountering an infected tick. So your risk is dependent really upon not only the infection rate, but your behavior, what personal protection measures you take, and whether you're doing tick checks. So at this point, I believe we're supposed to take questions related to what's been presented so far.
Yep, and we actually don't have any open questions, so we can carry on, but I do want to tell people if you do have a question, the way to do that is if you scroll your mouse over the screen where you're seeing the slides, you'll see there's a box that says Q&A underneath a couple of squares. If you click on that, you'll be able to ask a question, and then we will be stopping again in 10 minutes. I can ask any questions that come up there. So just wanted to remind people how to use the Q&A feature. So thanks, Kirby. That's very interesting. I was stunned that they lay 2,000 eggs. So do you know how many of those 2,000 eggs reach adult stage once they've been laid?
Fortunately, most do not. Most are not. That's a unique thing about a 3-host tick, is most of them are not successful in finding a subsequent host to proceed on development. In contrast to, there are a few tick species that are what we call 1-host ticks, and they can build up really large populations because they spend their entire life cycle feeding on one host animal. So little different biology there.
I was going to say with 2,000, you've got good odds.
Yeah, right. So let's get into how we're going to think about how we're going to manage our exposure to ticks or control them. And there's been a number of approaches that have looked into integrated tick management. Obviously, education and behavior change is important. Personal protection measures. I'll briefly go into landscape modifications. There's been a lot of research on chemical control, whether you're talking synthetic insecticides or newer, or essential oil, botanical-based or natural compounds. Work on biological control, host reduction or exclusion, host-targeted acaricides, and I won't have time to talk about it today, but there's also some research being done at looking at host-targeted vaccines. In other words, the idea is you would actually vaccinate the mice, which are our reservoir hosts that carry the bacteria, and render them incapable of infecting any ticks that are feeding on them. So let's look at our risk. We think that most of the risk here, at least in the Northeast, and contrast out in the Western US, where it's largely recreational, is right around your home. And this shows you the results of surveys that were done basically on ticks, either that were submitted for identification or testing. And you'll see that the respondents indicated, where did you pick up your tick? And most said outdoors right at home, 74%. 21% of the ticks were acquired in activities away from the home. What were you doing? Or what do you think you were doing when you picked up the tick? Well, interesting, play outdoors, 47% of the respondents, which is not too surprising, I guess, because if you look at the age distribution for Lyme disease, both in the Northeast and nationally, the highest incidence is actually in children, and the lowest incidence are those in their 20s. And then the incidence increases for people up through their 30s, 40s, 50s, and 60s, which would correspond with probably maybe home ownership and then increased activities. You see here, the risk of 12% was gardening, 18% was yard work. So we believe that, essentially your highest risk here in the Northeast for many people is right outdoors, as well as activities away from the home, like hiking. Your first line of defense is personal protection measures. Clothing, wearing pants tucked in the socks. I don't see people doing that very often, but remember, most ticks are down in the lower vegetation and are picked up on the lower extremities and crawl up. They don't fly, they can't jump, and they're not dropping from trees. So that's your first line of defense. You can use a skin-based repellent, deep picaridin, oil of lemon eucalyptus, but you want to use up in that 20 to 30% range products. 5% isn't going to do, maybe fine for mosquitoes, but it won't do much for ticks. There's also a permethrin-based clothing repellent, and a couple of Repel, Permanone are a couple of examples. This is a 0.5% permethrin. It's an insecticide. It's this material that's used on malaria bed nets. It's sprayed, as you see in the illustration here, onto the clothes, allowed to dry, and it can provide very effective protection. You can also purchase permethrin-treated clothing that's been pre-treated to last up to maybe 50 washes. And a recent study showed that it reduced tick bites by 58%. But I will say personally, I do use the permethrin-based clothing tick repellent. And over 30 years of tick research, I've had very, very few tick bites, and I actually have not gotten Lyme disease yet. So it can be very effective if used properly. Of course, important is bathing and doing tick checks when you come back, and promptly removing any ticks that you find. I actually prefer, given how small and nymphal deer ticks are, very fine tip forceps, grasp it as close to the skin as possible, and pull it out, pull the tick out. So let's get back to this residential or risk around the home. So management around the home can impact the number of ticks that you encounter in your yard. So here you'll see a map, correlate diagram of a residence. When you think about it, where the woods are, that's your tick zone. And the idea is to create a tick safer zone around your home. The majority of the ticks that I've recovered in the yard have been within 3 yards of the edge with the forest or stone walls or ornamental plantings, that kind of thing. Think cover, think pachysandra, think leaf litter. That's where the ticks are mainly found, and that ecotonnage. So you'll see in the diagram here in the upper left, a very kind of very shaded, wooded, unmaintained kind of area in the yard. The homeowners cleaned it up, and the number of ticks in the yard, even though they're still in the woods, was reduced by about 70% in that particular case. Removing leaf litter around the edge of the yard can reduce the number of ticks there by 49 to 70%, even though remember, they're still in the woods. Putting in landscape barriers can reduce the number of ticks moving into the yard by 35 to 77%. And of course, where do you often see the children's swing sets at many homes? Right back into the woods, which right back in the tick zone, where your risk of encountering a tick is quite high. So you really need to pull those out into more open areas of the lawn. But you know, I've talked about leaf litter, clearing it can help reduce the number of ticks. We found that leaf litter increases the overwinter survival of both Ixodes scapularis nymphs and Amblyom americanum adults. I'll be talking more about this in a subsequent webinar, but just briefly, I'm showing 1 year of a 3-year study where you can see where we removed leaves and snow versus where we did not remove any leaf litter or remove any snow. Snow is an insulator. And you can see we had 94% survival versus significantly lower 77% survival when you don't have that insulating leaves and snow present. However, another recent study found that leaf blown or raked accumulation of leaves at lawn edge was associated with increased numbers of nymphal Ixodes scapularis. So basically you're moving them from the area, the edge of the lawn where you've cleared them, to areas where you've piled them up. And as you can see in the graph here, the managed edge, a much higher number of ticks were recovered than along the natural edge or natural forest. So removing the leaves offside, bagging, possibly even a good idea, maybe composting them could help also reduce the risk there. At least don't let the children play in the whole pile of leaves. Another aspect of vegetative management is that invasive plants provide fantastic habitat for ticks. And higher tick counts and higher infection rates have been associated in the ticks with exotic invasive forest understory than in areas with native forest understory or open forest understory. So in studies here in Connecticut, the abundance of adult black-legged ticks infected with Borrelia burgdorferi was actually greatest in areas with dense stands of Japanese barberry. And similarly, there was larger numbers of Lone Star ticks infected with Ehrlichia in stands of invasive honeysuckle. And again, this is because it provides a good microclimate for the ticks and as well as some of the hosts. So reduction of both of these invasive plants reduces the number of infected ticks. Spraying continues to be one of the main lines for controlling ticks, particularly at many properties. This could be synthetic acaricides or insecticides such as carbamides. Pyrethroids is the main one that's being used. Neonicotinoids are in products some products for animals. Microbial biopesticides, Metarhizium anisophili under the product name MET52. This is a entomopathogenic fungus, which has also been used for tick control. And as you can see, there's a female tick killed by this fungus in some of the studies that I did. And there's a lot of current interest in botanicals and natural substances, which are essentially essential oils or plant extracts. And most of these are those that are listed on the EPA's 25B list of minimum risk pesticides. I'll say up front that these products come along and go, and it's hard for researchers to keep up in evaluating their efficacy. And I will say some work to some degree, and there are others that do not. So it's kind of like a Wild West right now in terms of which products are effective and how well they work. But again, some work and some don't. So what I've done in this slide, I've summarized some a review that's noted at the bottom there on research that's been done on the application of acaricides to the environment for tick control. You'll notice that the pyrethroid materials are quite effective, with control ranging up to 100%, lasting for several weeks. That's the top of the chart here, or graph, or table, I should say. And then carbaryl, which is a common garden insecticide, or Notradium 7, is also quite effective for a period of weeks to months. You can see that the biopesticide, Metarhizium anopheles, which is a suspension of the spores of this fungus, is also can provide variable, but potentially up to 96% control. Generally, I would suspect around 70%, lasting for a period of weeks. And there was one product early on that's been published, evaluated. It was a rosemary oil-based product called IC2. It's no longer available, but again, that was found to provide pretty good control. I did some trials with garlic. It's not a toxicant, it's a repellent, and it did knock down tick activity by roughly half for just a couple of weeks. So things to watch coming forward. We do have concern about whether Metarhizium anopheles, the MET52 product, will continue to be available. There's rumors running around about the potential sale or discontinue of this particular product. But we also did a lot of research, which has been published on Nucatone. Nucatone is a component of the essential oil from Alaska yellow cedar. You can also get it from grapefruit. And there's synthetic versions of it now available. It's a good repellent. It's a good toxicant. And there is a company that is developing this product as a tick control agent called NukaShield. It's currently under review by EPA. So it's just something to kind of watch going forward. It's not available yet, but we found that it was actually fairly effective in our research that we did on this product. Questions? Okay.
Okay.
All right. Yeah, I got it. I see it now. Yeah. Was the landscape barrier in the play set cedar mulch? Now, actually, it was a wood chip mulch, but it was not cedar. But there was a published study looking at cedar. And so the volatiles in cedar that were actually had some repellency against ticks, of course, volatilizes and only lasted, you know, a month or few months at the most, and then it was gone and wasn't any different than regular wood chips. Basically, you're creating a dry barrier that just minimizes the number of ticks crossing those substrates.
Okay, great, thank you. And if you have any other questions, please. Oh, looks like we do have one from Henry Murillo. Have you seen any major natural predators?
No, unfortunately, the tick has very few natural predators. There are, you know, there's some opportunistic predation that occurs on ticks, but it's not of any major significance. There is I did research for a while. There is a parasitic wasp that attacks the tick. Unfortunately, it's not very efficient, and it's only found in areas with tremendously high tick populations, and it tends to disappear when you have fewer tick numbers.
Great, lovely. Thank you very much.
So another area that a lot of research has been done is host-targeted tick control. If you think back to the life cycle of the tick, you know, it's feeding on various different host animals. And so particularly you're looking at the major host for the immature stages, like white-footed mice and chipmunks. Remember, mice are not only a major host for the immature stages of the black-legged tick, it's also the reservoir. The ticks actually pick up the Lyme disease pathogen from the mice and then subsequently infect a new generation of mice. So to just kind of simplistically, if we want to think about how Lyme disease exists out in the wild, the mice infect a new generation of ticks, the ticks infect a new generation of mice. White-tailed deer are not involved with the disease aspect of the cycle. They're the main reproductive host for the tick, and consequently, they're one of the major determinants on what your tick population is, not how many are infected. So you'll hear stories, oh, it's the mice, no, it's the ticks. It's both. I mean, excuse me, mice or the deer, but it's actually both that are involved in the entire life cycle. So there was work done on targeting these rodent reservoir hosts, such as the white-footed mice and eastern chipmunks, for Ixodes scapularis that you see here. The first product were these permethrin-treated cotton balls that were distributed in tick tubes that came out in the late 1980s. There's two products available, Daminex, and the Thermacell product. And the mice collect the cotton and take it back to their nests, and then they're exposed to the permethrin on the cotton, which would kill the ticks that are present on the mice. Chipmunks do not use the cotton in the tubes. And another more recent product is the fipronil bait boxes. These boxes allow access to both mice and chipmunks. It contains two non-toxic food blocks. They come in, there's a wick that contains fipronil. Fipronil is the active ingredient on frontline for flea and tick control on dogs and cats. It works very well on the mice. So there was just a recently published study, which I reference here, where they looked at these products individually. They found 27 to 20% control with the tick tubes in year 1 and year 2 of deployment. And they compared that with the bait boxes, where they've got 84 and 79% control of host-seeking nymphs on the properties in year 1 and year 2. Also bear in mind that these approaches will not work for Lone Star ticks, as the immature stages of that tick don't really feed them much on rodent hosts. The other approach has been white-tailed deer, and looking at from 3 aspects. So one has been research on exclusion, reduction, or the treatment of deer. So some studies that me and some colleagues did back in the early 1990s. In my case, it was a couple of properties that were about 12 to 15 acres in size that were surrounded by a 7-strand high tensile electric deer fence. Deer could not get in, and what you saw was a 100% reduction in larvae. You know, no female ticks, no larval ticks. There was an 84% reduction in nymphal tick numbers and a 74% reduction in adult tick numbers. So just excluding deer on a scale of acres can significantly reduce the tick population. Similarly, there have been a number of studies that have done looking at reducing deer populations to reduce the number of ticks. And typically deer densities in the Northeast are running around 30 to maybe 70 deer per square mile. It's very clear from a number of studies that you have to get the deer numbers down to around 10 deer per square mile to actually impact tick numbers significantly. But in the studies that have been done, there's been a couple that have shown, one study in particular actually was able to document some reduction in human tick-borne disease with the reduction of deer in one community. The last approach is the treatment of deer using this device called a 4-poster. It was developed by the USDA. It contains whole kernel corn, and it has 4 paint rollers that are treated with permethrin. So as the corn kind of trickles out in 2 little trays, as the deer come up to feed, they brush up against the rollers, and they're getting topically treated and killing any ticks that are on the deer. This approach can also be effective, and I've published a review of the use of these topical acaricides for the control of ticks on white-tailed deer in the Journal of Integrated Pest Management, and this is open access for those who want to follow up this in more detail. So what we have today is a tick-borne disease toolbox, and I've kind of highlighted a number of these. We have our personal protection measures. We don't have a human vaccine right now, although there is one that is being in phase 2 trials at the moment, a new Lyme disease vaccine, so we'll see how that goes. So perhaps in the future we will have another human Lyme disease vaccine. I briefly kind of highlighted the landscape and vegetation management approaches that can be taken. Killing host-seeking ticks on the property continues to be the main approach, but it ranges from the synthetic chemical acaricides to some natural product-based materials, as well as that nifty 52 fungal acaricide. Another area that we'll be doing research in the future is combining a insecticide or acaricide with a pheromone, an attractant, which would allow you to reduce the amount of pesticide that's actually being used. Rodent-targeted approaches went through briefly, mainly with the tick tubes and the topical acaricide bait box. I'm not talking about oral vaccine, but again, research is progressing, and there is a product that is under review by the USDA. It's an OSP-A, which is an outer surface protein A, same kind of approach that's used in the original human vaccine to vaccinate mice, and if that delivered through an oral bait. And we found some initial promising results with that. And if you vaccinate the mice, they can't infect the ticks that are on them. So we'll see how that develops going forward. And then, of course, we have our deer-targeted approaches by either topically treating the deer, reducing the deer fencing, and there's research trying to look into developing an anti-tick vaccine for the deer. What you'll note here is that those boxes indicate where some of these interventions have been used combined with another tick control method, and the stars indicate interventions where there's been some supporting data on reducing human Lyme disease cases, and both of those are deer approaches. So when you actually combine methods, you're talking about integrated tick management. So I'm not going to have time to go into a whole lot of detail on this, but I would refer you to two reviews, one that I wrote called Integrated Pest Management and Controlling Ticks and Tick-Associated Diseases, and another review by my colleagues Alexis White and Holly Graff on a review of tick control technologies for the black-legged Lone Star and American dog ticks. Both of these are published in the Journal of Integrated Pest Management, and both are open access. So I would encourage you to look those up for a good review of integrated tick management. But I will highlight a couple of studies here. This is one that we conducted here in the town of Reading in Connecticut. Both of these have been published. So what we did is we went in and had 4 1-square-mile neighborhoods. One was a control. One, we started doing deer removal, which we dropped after the second year of the study due to protests and interference by hunters, ironically. The third neighborhood was we used Met 52 and the bait box. And then the fourth neighborhood, we had the deer removal aspect, which was dropped, the Met 52 and bait box as well. And so what we found is that after 3 years, you can see here the number of ticks on the Paramiscus leucopus, the white-footed mice, the number per mouse on the control, the deer removal, which didn't meet its objective, so a little bit of reduction there. Deer, bait box, and Met 52, and then the Met box and Met 52 alone, which had significantly fewer ticks on the mice. And we actually reduced the questing nymphal tick population by 78 to 95% where we had the box and Met 52. And so we reduced the risk of encountering a single infected tick by 66% compared with no treatment by the third year of the study. We've initiated another integrated pest management study here in Connecticut in collaboration with the USDA in Maryland. And here we have 7 neighborhoods where we're looking at different combinations of the 4-poster device for treating the deer, the rodent bait box, and spraying with Met 52. And we did baseline sampling in 2017. We deployed the 4 posters, and all of the treatments have been applied in 2018, 2019, are continuing here in 2020, and I'll be able to report on the results of those in the future. And just kind of here, this just gives us a little more detail here. You can see that we distributed 10 bait boxes at the 9 properties within the 6 treatment neighborhoods. That's a total of 540 boxes. We purchased a spray rig to apply the Met 52, as you can see being applied here. And then we have 12 4-posters placed on land trust, town, and private property throughout the various neighborhoods in the community as well. So there's also a review of these deer-targeted methods, as I mentioned before, if you want to go into those in the Journal of Integrated Pest Management. But we have all these techniques I mentioned in the toolbox, and we're trying integrated approaches for trying to reduce the tick population. But it isn't going to work if nobody does it. So this was an interesting health style survey that was published back in 2015. It was a national survey, and I'm just focusing here on the Mid-Atlantic and New England states here. And in the one map there, you'll see that the percent of those people reporting an exposure by a family member to a tick in the past year. So New England, it was almost 30%. Mid-Atlantic, it was 24%. And then they asked what proportion or how many people were actually using a repellent, showering, doing tick checks, and other steps. And interestingly, here you'll see that in New England and Mid-Atlantic, you've got about a quarter of people that are reported actually using a repellent. Anywhere between 30 to 43% were doing tick checks. But surprisingly, you've got 36 to almost half of people reported doing nothing. Nationally, that was about half. And only a small percentage were using or applying yard pesticides for tick control. And then another proportion were declared that they would not use yard pesticides to control ticks at all. So I think as we try to increase our education on how on personal protection measures and various tactics for reducing tick populations, those numbers will hopefully go up. So I know we have a number of questions that have been coming in.
Yes, we do.
So all right. All right, Jill. So you think it would be better to have Nucotone used for people to prevent tick attachments? What's your opinion? Well, the Nucotone's a very good repellent, but right now it doesn't look like there's, the effort was focused on developing the product as a human use repellent, which is what I thought the original approach was going to be, as opposed to a, area-wide acaricide. Tick resistance does occur to pesticides, but that has shown up as a major problem more with some of the one-host ticks or in livestock applications where you get a lot of selection pressure. As you saw from the graph, a lot of people are not applying yard pesticides for tick control. So I don't really see a lot of selection pressure at this point. So that we'd end up with resistance to pesticides for Ixodes scapularis. Would Metarhizium anopheles target ticks only? What about other insects? The effect on non-targets is actually relatively small. There's been screening, actually there was a paper published with that concluded that the impact on non-targets was actually pretty low. It's a little selective. It doesn't really pose that much of a risk to beneficials and pollinators. This fungus normally occurs down in the soil. Some beetles are more susceptible to it. But again, I think the risk is minimal, particularly compared to chemical insecticides. Used with a soap in a shower bath in the evening would cause ticks to drop off, preventing it. Once ticks become attached, they are firmly attached. They're not going to wash off. You really have to physically remove them. They're not like mosquitoes that, with hypodermic mouth parts that come in, feed for 2 minutes and they're gone. A nymphal black-legged tick requires 4 days for full engorgement. A female black-legged tick requires 5 to 7 days for full engorgement before dropping off on their own. They actually cut into the skin and set their mouth parts. Most ticks actually secrete a little bit of a cement-like material to help hold on even better. And so you really have to physically use a tick removal device. Again, I use forceps, fine forceps, to grasp a tick and pull them off. If you have a high chipmunk population in your yard, what would you recommend? That gets a little tougher. The tick tubes will not work for chipmunks. The bait boxes would be effective for chipmunks because they're designed so that both chipmunks and mice will enter those boxes. So the boxes are an effective tool, but they are much more expensive than the tick tubes are. And they do require professional applicator, licensed applicator application. It's not a homeowner product, unlike the tick tubes. Another question, I recall from your group looking at a combination of MET52 and rodent bait boxes. Would you comment on that efficacy and cost to homeowners? Okay, so like I said, the tick tubes run about $3 plus a tube. The bait boxes, with the cost of the box, you actually use 2 each season. You put out the boxes to target the nymphal ticks in the early summer that are on the mice and chipmunks, and then in the later summer, another set of boxes to target the larval ticks that are on the mice and chipmunks, and they run about $49 a box, including installation. And I gave you some figures early on the efficacy of both of those. Short grass is often mentioned, but you can further define that. They recommend setting a mower to the highest setting. Is that considered to be short? I don't think so. You got to balance proper lawn maintenance. We just generally don't find ticks out in the open sunny portions of the lawn. As you do, again, mainly it's think cover. I have collected ticks in Pachysandra right next to the front doorstep where the garden hose is. And a lot of those treatments, even when those that do have acaricides applied to the property, is usually done as a perimeter spray, which is your highest risk area, but it's not your only area. So I think I've got just a few minutes. I'll give you an anecdotal example of a homeowner that was cooperating with one of my studies. She had a big circular lawn. I couldn't find any ticks immediately around the house, but I took a little short trip down her long, unmaintained edge and unpaved driveway to her mailbox and picked up 30 ticks. So she actually had gotten anaplasmosis, or what was then called ehrlichiosis at that time, and probably just in that going down to check her mail. Not right around the house, but so you really got to think about where your potential exposures are. But it's not going to be out in that open, sunny, manicured part of the grass, even at 3 to 4 inches in height. So yeah, the question on the bait boxes, Jill, yeah, they are pretty expensive, and I suspect a lot of people would have a hard time managing to pay for them. We actually did some, when the original study with those boxes was done on an island in Connecticut, we basically wiped out the tick population because all of the homes were treated in the initial study. And I actually would have argued at the time for a design that was actually more expensive. Perhaps a more permanent bait station where you would just replace the bait and the wick. But that's what we have. Why is a personal protection with Nucotone would be more effective, like a soap? Yeah, I think that might be a good approach. It is a very good repellent. But again, it's also very good as a treatment in the lawn as well. Although when they formulate the product, they will have to be careful because there is some phytotoxicity issues associated with it.
All right, and I think Kirby, maybe just one more question, and then we should close up the rest of the presentation.
Okay, so yeah, does greater diversity in the local host population affect the infection rate in ticks? It does to some extent, obviously. One of the arguments is greater host diversity would lower the infection rate in ticks, and what we have found that we actually do have a higher diversity, ironically, in woodlands right around residential areas than we do in our mature forests. But the mouse populations are similar in both environments. And so the infection rates are a little lower in areas where you have greater diversity, but you're not talking a really dramatic difference in the infection rate.
Okay, great. All right, wonderful. Well, thank you. That was incredibly helpful and informative. And we just have a few slides that we'd like to go through before we complete. So Kirby, if you could move it forward, that would be great.
Okay. Yep. Yeah, I didn't get to, we added a lot of questions, so I didn't get yet to the challenges to effective public tick control. We spent a lot of time answering questions there. I'll just go through these really, really quickly. The issues that we have, we're dealing with different tick species and ecologies. The big question is who's responsible for tick control on private property versus community and public lands or neighborhoods and school grounds. How can we deal with the low acceptability of many current tick control methods? And surveys have found a limited willingness to pay for them. Let's just go on. It's still unclear whether many of these tick control methods will actually prevent human disease. We know we can kill ticks, but actually reducing the incidence of Lyme disease and other tick-borne diseases is still remains a real difficult challenge. We don't have any municipal or local vector control efforts specifically aimed at ticks. I mean, some states have mosquito control districts. We don't have anything like that for ticks. We need more research on control of some of the species that are becoming of increasing concern. For obvious reasons, a lot of the research the past couple of decades or so has been focused on Ixodes scapularis due to the increasing Lyme disease issue, but we need to increase our investment and our research on Lone Star tick control as well, for example. How can we get industry to invest in developing new products for an unclear public health tick control market is a good question. And again, how effective are these materials when applied by homeowners or pest management professionals? The efficacy question is always coming up. So again, it's a homeowner problem, and most people largely rely on licensed commercial applicators. And you know, you come out and spray for ticks, but you know that doesn't really account for the huge variability that you find in risk around individual homes. So a lot of these issues were came up in the tick-borne disease working group and their various subcommittees looking at the broader tick-borne disease issue. And some of these also, you know, are highlighted here in this slide. And I think solving this is going to require a combination of public health agencies, the pest management industry, as well as the public. And Dr. Lars Eisen and myself just recently published a forum article on the barriers to effective tick management and tick bite prevention in the United States. This is also in the public domain, public access in the Journal of Medical Entomology, so anyone that wants to follow up this a little more closely can readily access this particular article. But in conclusion on this issue, it really comes down to how effective these approaches are. What kind of scale can we implement these things at? What is the cost of these interventions? And whether or not they're actually implemented will be key components for tick management strategies. So where do we go from here? Okay, widespread and difficult to control diseases from tick bites are a major problem worthwhile. We have a growing number and continuing spread of tick-borne diseases here in the United States. We do have a lot of tools available for killing ticks, but the impact of these on disease incidence is unclear or unproven, and a few methods are available or utilized by homeowners. We need safe, cost-effective, socially accepted, and effective prevention tools. And we do have multiple challenges or barriers to effective tick bite prevention, and we need to address those. So one approach is a One Health approach. Tick-borne diseases are difficult to control, partly due to their complex epidemiology and ecologies that involve different tick vectors and animal hosts. So basically, you got to think about the animal branch, the veterinarians, the veterinary entomologists, and wildlife biologists. And then what I call the human side. You got your physicians, the medical entomologists, such as myself, vector biologists, epidemiologists, and pest management professionals that really need to communicate with each other and come up with combined approaches to address this increasing health issue. Part of the solution is the fact that the CDC funded 5 universities with various partners that were established as regional centers of excellence in vector-borne diseases to help prevent and rapidly respond to emerging vector-borne diseases in the United States. And the Northwest Regional COE is based out of Cornell University. So that combined with our tick IPM methods that we're seeing like today are very important, I think, in addressing this issue. As I wrap things up here, I want to point out that there is a tick-borne disease working group. The charter for this group was approved by the Secretary of Health and Human Services back in August of 2017. And it was authorized by Congress for a total of 6 years from the date the act became law. The charter was defined, how the working group was structured and functioned. It was in response to the charge provided by the 21st Century Cures Act, and is renewed every 2 years in accordance with Federal Advisory Committee guidelines. The current charter expires in August of 2021. The first 2 years of the work of the group was published in a 2018 report to Congress. I encourage people to access this. It gives a really nice broad review of the whole issue, and work, subcommittee work continued on that as well. So that's an ongoing process. So I'll kind of wrap things up for any final questions here with this quote. I'll also want to mention that my tick management handbook was published in 2007, and it's available on the Connecticut Agricultural Experiment Station website. I'm in the process of updating it, but a lot of the basics that are in it haven't changed that much, so it's still a good resource. So we certainly have a lot more tools today than they had back in the mid-1800s. So I'll conclude with, from red bugs and bed bugs, from sandflies and landflies, mosquitoes, gallinippers and fleas, from hog ticks and dog ticks, from hen lice and men lice, we pray thee, good Lord, give us ease. Thank you.
Lovely, thank you. What a wonderful way to end it. So thank you, Kirby, very, very much for the work and the research that's gone into this presentation, and all the information contained in it. And I do want to share that Kirby has another engagement that he needs to run to. So we're going to do a quick switch of the screen, and we have some follow-up pieces for you. People were asking about the just hold on 2 seconds. There we go. People were asking about upcoming webinars, and I'll be covering that. So, so thank you very much, Kirby. I appreciate.
Okay.
I appreciate your.
I'll have to take off now. Talk to you later.
All right. Bye bye.
Okay, bye bye.
All right. So we have some questions for you, which are similar to the ones that were posted in the first poll. So if you would just take a moment to answer these questions for us, that would be fantastic. And then I'll be sharing with you about upcoming topics and presenters that we have coming forward for you. So I'll just be quiet for a minute while that happens. All right. So we've moved from moderately to somewhat knowledgeable, and a few more people in the very knowledgeable category. And that's the case on most of these questions. I did want to highlight the number of people who said that they're more likely to implement IPM at the end of this webinar, and that falls mainly under the very likely and extremely likely category. So we appreciate that. That's what we're after. Let me move forward and share some last things with you. If you are interested in connecting with colleagues such as Kirby in the Northeast around a particular topic, we have an option of a Find a Colleague on our website. You can go to this link and put in a profile, and also find other people who are working on topics that you're interested in. And these are the upcoming webinars that we have to date, and actually we've just added 2 new ones yesterday that didn't make it onto the slide yet, but we have Stephen Rich coming in a couple of weeks, talking about what happens when and if reducing the source or preventing tick bites has failed. We have Dina Fonseca and Matt Brickerton from Rutgers talking about Asian longhorn tick IPM. Alison Gardner from the University of Maine is going to be speaking with us about habitat management for vector-borne diseases. Doctors Jody Gangloff-Kaufman, Joellen Lampman, Matt Fry, and Laura Goodman, all from Cornell and the New York State IPM program, are going to be speaking about pathogens found in ticks collected on school grounds and public parks. And then we have host-targeted tick control, what works, what doesn't, and what's new, by Andrew Lee. And also Kirby is going to be coming back to speak with us in October about managing habitats in terms of leaf litter and snow removal, which he touched on today. So you need to register for each of those webinars individually, and we will be updating the website. There are a couple more people that we've reached out to, and I hope that we will include them in the mix. So please feel free to go there, register for each one of those that you're interested in today, and then come back in a couple of weeks and you may have a couple more options that are available to you. So, great. And just a reminder that there's a recording of this webinar is going to be available next week, and you should get an email into your inbox. And we don't want to end without acknowledging funding sources for this, which includes USDA NIFA through a grant that's provided by the Northeastern IPM Center, and also the Connecticut Agricultural Experiment Station that is where Kirby is employed. So, well, thank you very, very much for your attention, for your questions and your participation, and I look forward to seeing you on another webinar.